Case study of the day. Daniel. He was a healthy 35 years old man. Until one winter day when he slipped on some ice, fell forward and hit his head hard. The doctors cleaned the cut, ran a CT scan, no bleeds, no major fractures, and sent him home. But after a couple of days, he noticed something strange was happening, or rather something was not happening. He could no longer smell. Not his morning coffee, not his wife's perfume, not even the food cooking in the kitchen. In the weeks that followed, he burned dinner because he couldn't smell it started to burn. He couldn't smell the rain when he stepped outside or the gasoline when he filled up his car. He drank expired milk because he couldn't taste that it had gone sour. The world got a lot less interesting. Eating was not very exciting and Daniel started getting depressed. Life felt sterile and unfamiliar. Daniel had anosmia, a partial or complete loss of the sense of smell, and with it most of his ability to taste. This unfortunate condition can be caused by things like head trauma, respiratory infections, or even plain old aging. And I say unfortunate because what we sense informs who we are. The sense of smell is one of our 5 major special senses, and in this video we are going to break down how the system of smell works. And we are going to do that by first look at the anatomy of the olfactory system, the nasal cavity, the olfactory epithelium, and the specialized neurons that make smell detection possible. Then we will dive into the microanatomy of olfactory neurons. We will zoom into this area and see the structures of these unique sensory cells and the supporting cells that stabilize this system. While we are there, we will understand the molecular mechanism of smell detection, how odorant molecules bind to receptors and generate electrical signals. And then go through the olfactory pathway to the brain, take you through the pathway from the olfactory bulb all the way to the primary olfactory cortex, and also talk about why smell is uniquely tied to memory and emotions, the direct connections to the limbic system, the amygdala and the hippocampus, and why certain smells can instantly transport you back in time full of emotions. We'll also mention quickly how smell and taste work together and add some clinical correlations along the way, things like olfactory adaptation, anosmia, and hyposmia. What's up everyone, my name is Time, I'm a medical doctor and I make animated medical lectures to make different topics in medicine visually easier to understand. If you would like a PDF version or quiz of this presentation, you can find it on my website, along with organized video lectures to help with your studies. Alright, let's get started. Let's start by understanding where smell actually happens. When you breathe in, air enters through your nostrils and flows into the nasal cavity. Inside the nasal cavity, we can see these shelf-like structures called the nasal conchae, sometimes called turbinates. There are 3 on each side, the superior, middle, and inferior nasal conchae, and they increase the surface area of the nasal cavity to warm and filter the inhaled air. Surrounding the nasal cavity are the paranasal sinuses, which are air-filled spaces in the skull bones that drain into it. Here's the thing, most of the nasal cavity is lined with what we call respiratory epithelium. This tissue warms, humidifies, and filters the air you breathe. But there's a very specific region located high up in the roof of the nasal cavity. This is where you detect smell. This small yellowish region is the olfactory region. It's located specifically on the roof of the nasal cavity. This specialized area is only about 2 to 5 square centimeters on each side, roughly the size of a typical key on a computer keyboard. The reason this region is located up so high is actually very important because during normal, quiet breathing, some air does reach this area, but the airflow is relatively smooth and laminar. When you want to smell something more carefully, you sniff, right? And sniffing creates turbulent airflow that actively pulls more odorant molecules upwards into the olfactory zone. This increases the concentration of smell molecules reaching your receptors, which is why you instinctively sniff when you're trying to identify a smell. Now, you see the bone that forms the roof of the nasal cavity? This thin horizontal bone is called the cribriform plate. It is part of the ethmoidal bone, and the cribriform plate is special because it is actually perforated with tiny holes, about 20 to 30 small openings on each side. These holes allow nerve fibers from the olfactory region below to pass upwards through the bone and enter the space inside the skull. This is the only space in the body where nervous tissue essentially comes into direct contact with the outside environment through such a thin barrier. Remember Daniel from our case? When he fell and hit his head, the sudden impact caused his brain to shift inside the skull. The nerve fibers passing through these tiny holes in the cribriform plate can get stretched or torn by this sudden brain movement. This type of nerve fiber injury is one of the most common causes of smell loss after a head trauma. Studies show that 25 to 30% of patients with severe head injuries experience some degrees of smell loss. And often, there's no skull fractures at all. The stretching force alone is enough to damage these delicate fibers. And because this bone is thin and perforated, fracture here can create a pathway for cerebrospinal fluid to leak into the nasal cavity, or for infections to spread from the nasal cavity into the brain. That is why trauma to this region is taken seriously. Now, once those fibers pass through the cribriform plate, where do they go? They enter a structure called the olfactory bulb. The olfactory bulb is an oval-shaped structure that sits directly above the cribriform plate, resting on the floor of the anterior cranial fossae at the base of the frontal lobe. So now we've seen the anatomical landscape of everything related to smell, but what are the cells responsible for detecting smell? Let's zoom into the olfactory epithelium and see what's actually happening at the cellular level. What's really important to know about this first is that the actual olfactory epithelium is a type of pseudo-stratified columnar epithelium. This means the cells are arranged in what looks like multiple layers, but all of them actually touch the basement membrane at the bottom. They're just at different heights, which gives it a layered appearance. That is why we say pseudo, which means false. The olfactory epithelium is one of the two main parts of the olfactory mucosa. The other part is the lamina propria, the connective tissue layer underneath it. If we focus just on the epithelial layer itself, it is made up of three main cell types, each with a specific role. First, we have the olfactory receptor neurons, which are the actual sensory cells that detect smell. Second, we have the sustentacular cells, also called supporting cells, which provide structural and metabolic support. And third, at the base, we have the basal cells, which act as stem cells and continuously regenerate new olfactory neurons throughout life. Deeper in the tissue, located in the underlying lamina propria beneath the epithelium, we have Bowman's glands. These are specialized glands that produce the mucus layer covering the epithelial surface. Their secretory ducts extend up through the epithelium to deliver this mucus. Alright, olfactory receptor neurons. These are the cells that actually detect smell, and they're unique because they are bipolar neurons, meaning they have two projections extending from the cell body. One dendrite that goes towards the surface, and one axon towards the olfactory bulb. The dendrite that extends towards the surface of the epithelium, at the very tip of this dendrite, there's a small bulb-like swelling called the olfactory knob, sometimes called the dendritic knob. From this olfactory knob, 10 to 30 thin hair-like projections radiate outwards. These are called olfactory cilia, and they're absolutely critical for smell detection. These cilia are non-motile, meaning they don't move or beat like the cilia in your respiratory tract. Instead, they just extend outwards into the mucus layer that covers the epithelial surface, and it's on these cilia where the actual smell receptors are located. We will talk about this in detail in a few minutes, but an interesting thing with these olfactory receptor neurons is that they are one of the only types of neurons in your body that regularly regenerate. Most neurons, once they die, they're gone forever. But olfactory receptor neurons have a lifespan of only about 30 to 60 days. After that, they die and are replaced by new neurons. This constant turnover is essential because these cells are directly exposed to the environment, to pollutions, toxins, viruses, bacteria, and they take a beating, so the ability to replace them is crucial for maintaining your sense of smell throughout life. Now, surrounding and supporting these olfactory receptor neurons are the sustentacular cells, also called supporting cells. These are tall columnar cells that extend from the basement membrane all the way up to the surface of the epithelium. They are wider at the top and their nuclei sit higher up in the epithelium compared to the olfactory neurons. These cells, they not only provide structural support, but they also help maintain the proper ionic environment for neurons to function. And they metabolize. In addition to this, they clear away odorant molecules after they've been detected, so the receptors don't become overwhelmed. Without these supporting cells, the olfactory neurons couldn't function properly. You probably felt it during the COVID-19 pandemic where many suddenly lost their sense of smell. Initially, scientists thought the virus was directly attacking the olfactory receptor neurons, but research in 2020 and 2021 showed that SARS-CoV-2 actually infects the sustentacular cells, not the neurons themselves. The virus binds to ACE2 receptors, which are highly expressed on supporting cells. When these cells become inflamed and dysfunctional, they can't maintain the proper environment for the olfactory neurons, and smell detection fails even though the neurons are technically intact. In most cases, once the infection resolves and the tentacular cells recover, smell returns, though this can take weeks to months, and in some cases, the recovery is unfortunately incomplete. At the very base of the epithelium, we have basal cells. These are small, rounded stem cells that sit right on the basement membrane, and these are the cells responsible for that regeneration we just talked about. Basal cells continuously divide and differentiate into new olfactory receptor neurons to replace the old ones that die off every 30 to 60 days. This regenerative capacity is remarkable, and it means that even after certain types of damage, like a viral infection, the olfactory system has the potential to recover, as long as the basal cells themselves are not destroyed. Now, remember we mentioned the olfactory glands? These are specialized serous glands, and their job is to produce the mucus that covers the entire surface of the olfactory epithelium. This mucus layer is absolutely essential for smell because odorant molecules floating in the air are hydrophobic. They don't dissolve easily in water. But for these molecules to reach the receptors on the olfactory cilia, they need to dissolve in something. That's where the mucus comes in. The mucus produced by the Bowman's glands contains special odorant binding proteins. These proteins capture hydrophobic odorant molecules from the air and transport them through the mucus to the olfactory receptors on the cilia. The mucus also serves other functions. It provides a protective barrier for the delicate olfactory cilia. It keeps the epithelial surface moist, and it helps wash away odorants after they've been detected, so the receptors can reset and detect new smells. If your nose is dry, you literally cannot smell. This is true in the opposite way as well. When you're congested and your nasal passages are blocked with thick mucus, your sense of smell decreases dramatically. The odorant molecules just cannot reach the receptors properly. So that was basically all the cells at the olfactory region. So how does the olfactory receptor neurons actually detect a smell? Let's start with the odorant molecules themselves. These are small, volatile chemical compounds that evaporate from substances and floats through the air. For molecules to be smelled, it generally needs to be small enough to become airborne, and it needs to have the right chemical properties to interact with our receptors. We are talking about molecules from anything, things like food, coffee, flowers, gasoline. Anything with scent releases these odorant molecules into the air. When you inhale, these odorant molecules enter the nasal cavity and dissolve in that mucus layer we just talked about. The odorant binding proteins in the mucus captures these molecules and transports them to the olfactory cilia, where the actual detection happens. Now, the actual detection happens through odorant receptors, which are specialized proteins embedded in the membrane of those olfactory cilia. These receptors belong to a family called G protein-coupled receptors, and the discovery of these odorant receptors is actually one of the landmark achievements in neuroscience. In 1991, two scientists named Richard Axel and Linda Buck identified the genes encoding these olfactory receptors, and they were awarded the Nobel Prize in 2004 for this work. What they found was that humans have approximately 400 different functional odorant receptor genes. This makes it the largest gene family in the human genome. Interestingly, we actually have about 1,000 odorant receptor related genes total, but around 60% of them are pseudogenes, non-functional remnants from evolution. Other mammals, especially dogs, have many more functional receptors, which is why their sense of smell is far more sensitive than ours. Now, each olfactory receptor neuron expresses only one type of odorant receptor. This is called one neuron, one receptor rule. So out of the 400 possible receptor types, each individual neuron picks just one and sticks with it for its entire 30 to 60 day lifespan. But here's the clever part. A single odorant molecule can activate multiple different receptor types. The brain interprets smell not from one receptor firing, but from the patterns of which combination of receptors are firing. This is called the combinational code. With 400 receptor types, the number of possible combinations is astronomical. In fact, a study published in 2014 estimated that humans can distinguish over 1 trillion different odors, far more than the old estimate of 10,000. So even though we only have 400 receptor types, the combinational nature of this system allows us to detect almost unlimited variety of smells. Let us say you just walked past a bakery and the molecules from freshly baked bread enters your nose, dissolves in the mucus, and drifts over to the cilia of an olfactory receptor neuron that has a receptor that is responsive for that molecule. Step 1. The odorant molecule binds to the odorant receptor on the ciliary membrane. The receptor is a G protein coupled receptor with 7 transmembrane domains forming a pocket where the odorant docks. Step 2. When the odorant binds, it activates G protein on the intracellular side. This specific G protein is called GOLF. The olfactory G protein is a variant of the stimulatory G protein family. Step 3. Once activated, GOLF exchanges GDP for GTP and dissociates. The active subunits then activates an enzyme called adenylyl cyclase type 3, which is highly expressed in olfactory cilia. Step 4. Adenylyl cyclase 3 converts ATP into cyclic AMP, and cyclic AMP levels inside the cilium rapidly increases. Step 5. The rising cyclic AMP directly binds to and opens cyclic nucleotide-gated ion channels in the ciliary membrane. These channels are non-selective cation channels, meaning they allow both sodium and calcium ions to flow into the cell. Step 6. The influx of sodium and calcium depolarizes the membrane slightly, and the calcium that enters the cell then activates calcium-activated chloride channels. Now, here's the interesting part with olfactory cilia. The intracellular chloride concentration is unusually high. So when these chloride channels open, chloride actually flows out of the cell, which further depolarizes the membrane. Step 7. If the depolarization reaches threshold, an action potential is generated. This entire cascade from the odorant binding to action potential happens in milliseconds, and this system is extraordinarily sensitive. Studies have shown that olfactory neurons can respond to just a few odorant molecules. One unique feature of the olfactory system is how rapidly it adapts to continuous stimuli. You've probably noticed that when you first enter a room and you smell some food cooking, but after a few minutes, you stop smelling it. This is not because the smell went away. Your olfactory system has adapted. The adaptation happens through multiple mechanisms, one of which is because the calcium that enters binds to calmodulin, which together reduces the sensitivity of cyclic nucleotide-gated ion channels. This negative feedback quickly desensitizes the receptor, allowing you to detect new smells rather than being overwhelmed by constant background odors. So, an action potential has been fired. That signal now needs to get to the brain and be processed. Let's follow that pathway. The axons from the olfactory receptor neurons are thin and unmyelinated, and they bundle together in small groups of 10 to 100 fibers. These bundles are called olfactory filaments, and they pass upwards through those tiny holes in the cribriform plate to the olfactory bulb. Inside the bulb, the incoming axons synapse with second-order neurons in structures called glomeruli. All olfactory receptor neurons that express the same odorant receptor type converge into the same glomerulus. So if you have, say, type 1 neurons that all express the same receptors, every single one of their axons will target the exact same glomeruli. Another type of neurons that express the same receptors will target another same glomeruli, and so on. Within each glomerulus, the axons synapse onto 2 types of projection neurons, mitral cells and tufted cells. These are the neurons that will carry the signals out of the olfactory bulb towards the brain. Each mitral cell sends a single primary dendrite into one glomerulus, where it receives input from thousands of olfactory receptor neurons. There are other accessory cells here in the olfactory bulb as well that regulate the signals and sharpen the timing of the signals. We did cover them in the video about the actual olfactory nerve, but for now, let's follow the axons of the mitral and tufted cells as they leave the olfactory bulb and forms the olfactory tract, which runs along the undersurface of the frontal lobe. As the tract extends backwards, it reaches an area near the anterior perforated substance, where it widens into a triangular region called the olfactory trigone. And from this trigone, the tract splits into 3 separate pathways: the medial olfactory stria, the lateral olfactory stria, and sometimes an intermediate olfactory stria. The lateral stria is the largest and carries most of the olfactory information. It travels to several regions collectively known as the primary olfactory cortex. This includes the piriform cortex, which is the largest component and sits in the temporal lobe. It also goes to parts of the amygdala, specifically the cortical nucleus, and to the entorhinal cortex, which is the gateway to the hippocampus. And here's where things get really interesting. Those fibers project directly to these cortical and limbic structures without passing through the thalamus. The thalamus sits deep in the brain that is basically responsible for taking in sensory information and directs that sensory information to the cortex for conscious perception. This makes olfaction unique amongst all the sensory systems. Every other sense, vision, hearing, touch, taste, they all go through a thalamic relay before reaching the cortex. But smell, it bypasses the thalamus entirely and goes straight to the emotion and memory centers. Now, for conscious perception and identification of smells, when you can actually say that's a coffee or that smells like roses, the primary olfactory cortex actually sends projections through the mediodorsal nucleus of the thalamus to the orbitofrontal cortex. So there is a thalamic pathway, but it's secondary, not primary. The direct pathway to the limbic system happens first, and that is what makes smell so uniquely tied to emotion and memory. So we've just seen that smell has a direct connection to the amygdala and the hippocampus, your emotion and memory centers. You have probably experienced this. You smell something, maybe a particular perfume, the smell of rain, or the scent of old books, and suddenly you're transported back to a very specific moment in your past. Maybe your grandmother's house, your first day of school, or a summer vacation. And it's not just a vague memory, it comes with the full emotional weight of that moment. This is called the Proust effect, named after the French writer Marcel Proust, who famously wrote about how the smell and taste of a cookie dipped in tea brought back an intense flood of childhood memories. And this isn't just poetic, it's neuroscience. Research have consistently shown that olfactory memories are more emotional, more vivid, and more resistant to forgetting compared to memories triggered by other senses. Brain imaging studies show that when people recall memories triggered by smell, there's a greater activation in the amygdala compared to when the same memories are triggered by visual or verbal cues. Why is this? It comes back to that unique anatomical pathway. Visual, auditory, and tactile informations all pass through the thalamus before reaching the cortex, and only then do they connect to the limbic system. But olfactory information goes directly to the amygdala and the hippocampus with minimal synaptic delay. Smells get tagged with emotional significance immediately at the earliest stages of processing, which is why they are encoded so deeply. From an evolutionary standpoint, this makes perfect sense. Smell has been critical for survival for millions of years. You need to remember which foods make you sick, which plants are poisonous, which locations are dangerous. Attaching strong emotional weight to smells ensured that those memories stuck. Now, we need to address something that ties directly into Daniel's experience, the relationship between smell and taste. Because when Daniel lost his sense of smell, he also lost most of his ability to taste. And that's not a coincidence. Taste, or gustation, refers specifically to the 5 modalities detected by taste buds on your tongue: sour, salty, sweet, bitter, and umami. Those 5 sensations are the only things your tongue can actually taste. Everything else, all the complexity, the difference between strawberry and raspberry, between coffee and tea, that's not taste, that's smell. When you put food in your mouth and chew, you release volatile aroma compounds. These molecules travel from the back of your mouth up through the nasopharynx into the nasal cavity from behind. Studies estimate that about 80 to 90% of what we perceive as taste is actually smell. Your tongue can tell you sweet, slightly sour, some saltiness, but your nose is telling you this is a strawberry, it's ripe, it's fresh. The brain integrates these signals in the orbitofrontal cortex to create the unified perception of flavor. And this is exactly what happened to Daniel. After his head trauma, he had anosmia. In many cases, the taste buds themselves remain largely intact, but without smell, food had no complexity, no richness, no flavor. This is why patients with anosmia often report significant weight loss and depression. Eating becomes a purely functional act instead of a pleasurable experience. So when we talk about the sense of smell, we're not just talking about detecting odors in the environment. We are talking about a system that is fundamental to how you experience flavor, how you form memories, and how you navigate the world around you. And with that, we've covered all the 5 basic special senses in the previous 5 videos. I really hope you found that helpful. I've made free courses on other topics here on YouTube if you want to keep learning. Otherwise, if you want a handmade PDF version of this lecture, take a quiz to test your knowledge, or access an organized list of all my videos, you can find everything on my website. Thanks for watching. See you in the next one.